what you find is that when you look at these effects of antenatal maternal depression or socioeconomic status on volume of the right amygdala, right hippocampus, or the prefrontal cortex, is you find that there are different biological pathways mediating those effects. In the case, for example, of the impact of maternal depressive symptoms on the right amygdala, it is mediated by a glutamate receptor activity syndrome. That is the network right here. So that SNPs within the genes within this network are overrepresented and account disproportionately for that interaction. They involve genes that mediate glutamate receptor signaling, and they involve the snare complex, which is a complex of genes that is involved in synaptic formation, right? Well, that little baby pops up in 4 of the other networks as the single top biological function. So what it's telling you essentially is that if you want to understand the biology of how antenatal maternal depression is influencing brain structure in utero, is that it involves different neurotransmitters or neurotrophin systems operating at different parts of the brain. I don't think that's a big surprise. That they are coupled to specific intracellular signaling, most of which are involved with shunting metabolic resources to one part of the cell, and that they are then coupled to variations in a complex of genes that are mediating synaptic plasticity. So what you've done, essentially, is to go back to your GWAS data, hunt, and find biological pathways that then link your environmental condition of interest to your outcome. What genes allow us to do is get inside the head and perhaps even start to understand how different biological pathways are mediating these G by E's at different levels of the brain. So, this I think is really a little bit at the front end. But I'll give you one that I think for those interested in neuroendocrine function is kind of fun. You can take this also to a slightly different level of analysis. We actually did, with only 189 subjects, we ran a GWAS. We ran a GWAS against different brain structures. And the one that I'm going to illustrate here is the amygdala. This is diffusion tensor imaging, so it's talking about microstructure within the amygdala. And we actually even had some SNPs that passed false discovery rate. We didn't take them too seriously, but what we did do was to say, okay, let's take the top 100 polymorphisms in this GWAS. Those polymorphisms most statistically associated with variation in amygdala microstructure at the time of birth. And then let's ask, if we map those polymorphisms onto genes, do they form a coherent network that might then be associated with variation in amygdala structure? And the answer is they do. And look what sits at the hub. It's the glucocorticoid receptor. Well, I can't tell you the joy around the lab when we found that little baby. But this is the network. I don't know why I'm pointing to my screen like somehow I'm going to draw you into this. This is the network that describes a biological system that is most closely implicated in establishing variation in amygdala microstructure at the time of birth. And as you see, the glucocorticoid receptor is sitting at the hub. You can do the same thing with the hippocampus. Here you find a gene CUX1, which is not well described, but is clearly involved in synaptic plasticity. And you can do the same thing with the gene environment networks, et cetera. And again, you find that the glucocorticoid receptor and the CUX1 are also involved in mediating these particular pathways as well. So essentially, what we've got is this massive challenge. Right? We can't do what the people in diabetes do. We can't take abdominal fat biopsies. We can't take muscle biopsies to study insulin resistance. We can't take blood samples and look at platelet diffusion and coherence to look at the risk for atherosclerosis. We study the brain. But potentially what we can do is to start to think about taking advantage of all this GWAS data that's emerging, and particularly the GWAS, so as Dr. Bradley was talking about, when you get to doing GWASs on very specific conditions, then we're going to end up, I think, being able to accelerate our studies. But those studies, when they involve the integration of genomics, systems biology, and neuroscience, I think could take us someplace that would be really interesting in allowing us to get at this question of links. How is it we can understand these links? Now, you may turn around and say, well, you know, understandably, this is great that you identify some of these funky-looking brain networks, but how is that going to give me a heads up on what to look for in a child population? Well, I'd argue, I'll bet you that Andre and a lot of the work he did on neuroimmune was in fact inspired by the fact that people like Carla Schatz and others who did the early pioneering work on gene expression. 2 minutes, actually perfect, I only have 23 slides left. That when they did this early work, immunological genes popped up as they're expressed in the brain and highly associated with synaptic plasticity. So it may well be that our challenge now is to go back and start seeing, can we find peripheral signals that associate with these systems within the brain? I recognize this as a challenge. We've got a number of challenges. The brain, of course, as we say, is not easily studied. We can't take brain bioassays. Postmortem analysis only takes us so far. So that's one of the reasons for our enthusiasm in being able to use genetics to understand potentially candidate biological systems that mediate the impact of these environmental effects. We have a big challenge here. Major psychiatric disorders show early onset. The folks in heart disease, they can linger a little bit, right? I mean, they're looking at diseases that largely affect people by midlife. Detection isn't quite as pressing. And when you start working with high-risk individuals, you can get participation of those individuals in biological samples that we can't get, right? In many cases, access to these type of populations is difficult. Getting biosamples is difficult, and there's a rush, right? The major age of onset of psychiatric disorders is late childhood, early adolescence. So we got to get on it. The prevention target is obviously then children and early childhood, which means that we need to access our populations in clinical settings that are often quite difficult and challenging. We need to get in and work with pediatricians. We need to work with schools, et cetera. So we've got a lot of challenges, but essentially we've got a pathway at least built out to us. And just my congratulations to you folks here at Penn State for having marshaled a program that really does have the potential to take us down this particular list. Thank you very much.
Great. Thank you very much, Dr. Meaney. At this point, what I'd like to do is invite our previous speakers from the session to come up so that we can have a bit of a discussion as well as some questions from the audience about the session at large. Questions? Can we eat now?
Not quite yet.
Not yet. That's all right.
Sorry, guys. One question at least before we go to lunch. But great set of talks. Really just amazing work. I've enjoyed learning from all of you. And this question could be for any of you, really. I wonder, particularly in looking at peripheral epigenetic markers, if we're artificially limiting ourselves in a way to those mechanisms that may cross the blood-brain barrier. And I know, Michael, you're sort of touching on this issue. How do we think about these peripheral markers and what they mean for the central nervous system? And I wonder what you guys think. Are there mechanisms we may also be missing in this process that aren't necessarily reflected in the periphery, rather?
Yeah, I'll give a short answer. I'd say yes, you're absolutely right. But I think we may have underestimated the degree to which peripheral systems are implicated. So one of my sources of inspiration is some work done by the Toth group showing that polymorphisms in the TPH1 gene are highly predictive of behavioral outcomes in mouse models. TPH1 is not expressed in the brain. It's TPH2 that's expressed in the brain. TPH1 is responsible for serotonin only within the periphery. So I think you're right. I think you're absolutely right that we will miss some things, but it may be that there's more to be had there than we originally suspected. So half full, half empty.
And I think I would just follow up on that point in that, you know, that there's at least a couple of studies now that have shown that genetic influences on DNA methylation may be conserved across peripheral and central tissue. So, for example, the COMT polymorphism, also polymorphisms in D or D4 have been looked at in lymphocytes as well as in postmortem brain tissue, and you do see the same kind of genetic influence across the tissues. The question is then whether or not you see the same G by E effects, and the work on FKBP5 suggests that perhaps you may see those effects across the tissues. But you're right, I guess there will be stuff that we won't be able to assay in the periphery.
Yeah, and why do you think that is, that we're seeing similar epigenetic changes in the periphery and the brain? I mean, do you think it's related to, many of these are glucocorticoid sensitive regions? Are there other mechanisms at work that would be affecting the entire organism?
Yeah, well, at least in the case, and Bekh and Elizabeth Grinder would be able to answer this better in terms of FKBP5, but I think one of the roles that these polymorphisms is playing is influencing the 3D structure of the DNA, and that's influencing the accessibility of transcription factors to the underlying regulatory regions. So I think the structural effects of these SNPs is probably influencing the capacity of these factors to bind, including the factors that then regulate methylation, such as the DNMTs and the TET enzymes. So I think by changing the structure of the DNA or the accessibility of the DNA, then you may see common effects across tissues, but that will also depend on those tissues being expressed, or those factors being expressed in those different tissues.
If I could just chime in relation to telomeres. So many of the open questions in telomeres are very similar to what you have in epigenetics. Obviously, we are limited when we doing this kind of studies in humans. We don't have access to brain tissue. But if you think about the relationship between stress and health and the involvement in telomeres, there is a specific tissue, specific cell that seems to be more important. And many people are now trying to look at specific types of cells. There's some indication that T cells are more important. At the end of the day, if you look at telomere, and most of the people look at telomeres and leukocytes, you get an average measure of a very heterogeneous group of cells. And we don't really, and we know there is a specific erosion rate in specific types of cells in leukocytes. So it can be really important to understand a specific cells effect.
Thanks.
Could any of you speak to the role of 5-HMC, especially seeing as how that might be more specific to the brain instead of the periphery, and then how we might translate 5-MC in the periphery to possibly 5-HMC in the brain?
Well, having spent, we do a lot on 5-HMC. It's not clear how it's going to relate to the periphery at all. It is found, and I think there's certain cellular populations. I wouldn't be surprised that immunological cells show some variability that may co-vary along that particular line. Certainly, the enzymes responsible for the catalyst of 5-hydroxymethylcytosine exist and are dynamically regulated. We just simply don't know. I think, this is just a speculation, but I think where we're more likely to find, in the brain, for the most part, 5-methylcytosine and 5-hydroxymethylcytosine patterns overlay each other pretty closely. And I think 5-hydroxy is very informative, probably more informative than 5-methylcytosine, but the fact that they overlay may not be really, it may suggest to us there isn't that much more to be learned. That's a strange statement, but especially since we just spent hundreds of thousands of dollars doing 5-hydroxymethylcytosine assays. I think where you're going to see more is with non-CpG methylation, so-called CPN, CPH, that is where the cytosines are methylated but not in partnership with the guanine. And there's a lot of variability occurring in brain that I think does map onto peripheral tissue. The array that Kieran talks about does have a little bit of those marks, and there's not enough to really make any conclusions, but there is some. I think that's where we're going to see, because I think that's a more dynamic signal in the brain. There's some early evidence that factors such as environmental enrichment produce greater variability in CPN methylation than they do in classic CPG. So I have the feeling that's going to be an area that is not, by definition, going to overlap with classic 5-methylcytosine patterns. And that will give us something that I think is going to be a sandbox that's going to give us a lot more information. 5-hydroxy, it's interesting, but I'd be more optimistic that 5-CPH is going to be the mark that's more informative.
Is there time for one more?
Speculation.
No? Is there time for another question? This will have to be a naive question because that's the best I can do with genetics and epigenetics. But I know that when the new DNA is created at birth, at conception, almost all the epigenetic changes are wiped clean, and we're back to genes. But some are not. And given the preponderance of evidence for the epigenetic modification of the cortisol receptors, the GR receptors, is there any evidence that can be transmitted across generations with the creation of new DNA?
The glucocorticoid receptor is your thing.
Yeah, I guess. Not at the glucocorticoid receptor. There's certainly increasing evidence that there are sites that are protected, acting like classic imprinted genes. There's no evidence suggesting the glucocorticoid receptor can be directly transmitted. I think so.
And I think.
Oh, I'm sorry. Go ahead, Kieran.
Well, I was just going to say, as you correctly pointed out, almost all of the marks are wiped out, but not entirely. So I think there is the possibility for sustaining transgenerational effects, but the evidence for their existence in human studies so far is there's not a whole lot out there at the moment.
I think that we're done with this,
We are
right?
done. Yes.
Okay. So thank you all. And let's give a little hand here. Thank you. And thank you, Chad, for moderating this difficult session. Okay, now here comes the challenge. The challenge is for all of you to get your lunch in a timely manner and come back, but there's another very large challenge. The challenge is that we moderate our own behavior a little bit. At the end of today, we have 45 minutes set aside where we have a panel of our afternoon discussants and our integrative speakers throughout the 2 days coming up here to form a panel. You have to understand that this is an extremely rare occasion that all of these people from all of these different fields are together on one stage. What we want to do, this conference is devoted to moving science forward and to translation of how this knowledge can be useful for workable strategies for kids. So I want us all to think about our questions during that panel discussion. We were pressured to make this a webinar. We decided we did not want to make this a webinar because we wanted this to be a more intimate gathering. There's an advantage of a webinar, and a webinar, the only advantage is that you have people who have questions who type those questions into a computer, and then you get to go through those questions and sort of pick the questions that are most useful to the mission of the meeting. So we're going to do a version of that here. Instead of having everyone stand up at the microphone, we're actually going to go through the questions a little bit. There are 2 ways you can submit a question for the end panel discussion. First is by texting this phone number here. Okay, make it very succinct and to the point, and we will transcribe those so that we can go through those. The second is, if you don't have the technology, that's fine. Please clearly write your question on a piece of paper and give it to one of these 2 individuals here, Carlo and Erica. They'll be sitting there and they will help transcribe those. And then we'll be able to go through those questions as the afternoon session goes along, and as you eat lunch and you think of your questions, let's compile those so that we can take full advantage of these people's time, all right? And the group of you that are here that are on the translational fronts to move this science forward. So I'm hoping that we're all agreeable to that and that we'll see how that panel discussion goes, okay? So go off and have lunch, and I'll challenge you to get back here at 12:30. Thank you.